All for coming this afternoon. We're delighted to have Eric Engstrom from our biology department speaking. Eric earned his BA in biology from Reed College and his PhD in biological science from Stanford. And his PhD was on cellular physiology, signaling, and modulation. He was a postdoc at UC Davis, and he was a professor at William & Mary and Rollins College before coming here in 2005. And we're 15, I thought. I can't read my writing. So.
Yes.
Well, you know, on geologic timescales, 10 years is nothing. So, we're delighted to have Eric talk to you today. Thank you.
Thanks very much, Chris. Yeah, it's 13. I started teaching college in 2005. That's where the 13 years comes from. Actually, I made this title up some weeks ago when Lori asked me for a title, and at the time I was thinking this was going to be a little bit more of a sort of chronological progression. It turned out to be a lot less than that. It's more focused on the now, but I didn't change the title because I didn't want to cause confusion. And this is my research group, much of it, not all of it, from last year. They also featured very prominently in the first edition of this, and then they kind of fell out. But I can't bear to take them off the title slide, so there they are. That's Daniel, Caitlin, Alana, and Maddie. So this is a slide from the colloquium that I gave here about a year ago. The subject of that colloquium is sort of encapsulated in the slide title here. Is ethylene even a hormone? And what I wanted to do in that colloquium was sort of set up the framework, the questions that I'm asking in my research, the work I'm doing with my students. Gesundheit. And it revolves around this idea. So if you, I'm going to quickly go through this to kind of pick up where I left off, and also because ethylene will be coming back a little bit in this talk. So if you pick up any textbook in plant biology or plant physiology, there will be a chapter that will tell you that ethylene is a very important plant hormone. Okay? It's one of the big five, the classic ones that we know of, along with auxin, cytokinin, gibberellin, and abscisic acid. Okay? And we've thought this for many, many decades now. Okay? But the argument that I was making there was, if we take what we know about the chemical properties of ethylene, if we take what we know about the physical properties of ethylene, if we take the information that we have gathered in recent years about where ethylene is perceived in the cell, this idea that ethylene in of itself is a hormone doesn't exactly fall apart, but it becomes a little problematic. And a lot of questions start bubbling up. Okay? So it's incontrovertible that if you take any green plant and you gas it with some ethylene, it's going to show a growth response. That will happen, I guarantee it. It's not arguable that if you take a green plant and you put it in a box and you measure the atmosphere, that plant is going to be generating ethylene. Okay? That happens too. But after 100 years or so of working and thinking about this, we cannot demonstrate anywhere that there's a particular environmental input that causes ethylene to be produced in a particular part of the plant, that the ethylene then goes to a different part of the plant and initiates a response, an adaptive response. We can't put that whole story together anywhere, okay? And so most of the questions that we're doing in a research-wise have to do with sort of challenging this idea and coming up with an alternative idea for what ethylene might be doing, okay? And that's where I left you, those of you who were here for my last colloquium. What I want to do today is switch gears just a little bit, actually. I am going to talk about my research. I'm going to talk about work that my students are doing. But I want to talk principally about how I've been integrating my teaching and my research together. Okay? So that's going to be the major theme here today. So, I discovered that it's very easy to put a research talk together. They write themselves, and that's all I've ever done. You have the background, which leads to your question, which then leads to your experiments, and then your results, and how do I answer my question. This is just so easy peasy. This is the first time I've ever tried to give a talk that really talks about my teaching quite a bit. And putting this in a really nice linear argument absolutely it defied me. I couldn't quite do it. So I'm giving you forewarning here. Normally I would have a nice outline of where we're going. Instead, I'm just going to tell you about the sort of questions and themes, and this is going to be, and we're going to bounce around a little bit here, forewarned. So I'm going to talk principally about two courses. First, Bio 202 Genetics, okay? Thanks for coming, guys. Extra credit, yeah. And also the Seepage Laboratory, which is a new thing we are just starting up this semester. This is our current Seepage's cohort. That's Tim Tibbetts smiling back there, okay? So these two courses, mostly we're talking about 202 Genetics today, okay? 3 questions are going to form the framework for the first half of this talk, more or less. What do we want biology undergraduates to learn? Okay? I don't know how deep I was into teaching college before this question first popped into my mind, but longer than I am comfortable telling you. Are there best practices for teaching undergraduate biology? Okay? Another interesting question. How do we create conditions for undergraduate biology students to fall in love, and I want to emphasize with biology, not with each other. Okay? And that actually sounds kind of silly, I realize, and I actually feel a little silly putting it up there. It sounds like I'm proposing we make nice food for them and play very white music. But what this entails is motivation. It's the idea that this is hard work. Biology is hard work. There's a crushing amount of things you have to learn before you can call yourself an expert in biology. And you don't do hard work. None of us do hard work. No rational creature does hard work unless you have a good motivation. And when you fall in love with anything, it becomes a bit of your identity. You know, that person, that thing, a little bit of who you are. And that's a motivation that's going to draw you back time and time again. That's going to keep you humming in the library. And this came very late to me, I think, really, in those 13 years, was this idea that, you know, we're not covering material. We have to somehow set this up so that those people who might, you know, fall in love with this subject will and can. Okay, not everyone will. Not everyone will. Okay, so those are the 3 questions we're going to go on. I want to define some terms that I'm going to talk about a lot up front here. So I'll be referencing these ideas again and again. The first one is course-based undergraduate research experience, or CURE. I didn't write that acronym. It's out there in the world. So what is a CURE? This is a lab. This is, or rather, an alternative to a traditional lab. CURE engages students in ongoing, authentic research in the context of a course-affiliated laboratory. Everybody ever had a science course in this room? Yes. And there's always a lab, right? You know, I'm going to lab. Okay. This is what you do in that lab section of the course, an alternative thing. What is authentic research then? This is my own definition. Authentic research is research on a significant question. That is to say, not only something that we don't know, for which the answer is not known, but something that is worth knowing, you know, something that the discipline would actually care about, something that's going to really move us forward, okay? Which employs state-of-the-art methods and equipment and generating publication-grade data, okay? Doesn't mean that the data is going to be published necessarily, but that's the nature of research too. Most of the data that's in all of the notebooks I've been accumulating over the past several decades has not been published, okay? But I'm trying always to get publishable, publication-grade data, good stuff. And this is the goal for authentic research as well. Okay? And then there's this thing, student-centered learning or active learning. I use these terms interchangeable, and I think that's completely legitimate. I don't see, when I go into the literature, any conceptual difference between using one term or the other. How is this defined? This is a quote from this paper, which I think made the most sense to me when I read it. The paper is actually about teaching science to middle school students, not to college students. But it's about active learning. And early in the paper, they're defining their terms here. This approach to science teaching will require a shift from the teacher-centered instruction common in science classrooms to more student-centered methods of instruction. The defining feature of these instructional methods is who is doing the sense making. Who's constructing the knowledge? Who's putting it together? The instructor for the students, or are the students synthesizing that understanding? Okay, that's the basic idea. That's what's active about it. Okay, that's what's active about active learning. It doesn't imply that they're necessarily standing or using the blackboard or something like that. It's the construction of that knowledge, okay, who's putting it together. All right, so we've established now, it took some doing, but I got here in 2015. And the first course I taught was genetics, okay? So this is the beginning. How did I teach it? I taught it very conservatively, okay? Which I thought at the time was befitting someone who just gotten here. You know, you don't rock the boat too much. You see, well, what have we been doing here? So I taught this course as near as I could tell the way it had been taught in the previous year and the year before that and the year before that. And this is what it looked like, okay? There was a lecture component, all right? I did not have a picture of me lecturing to students in 2015. I rather like this. Comes from a 14th century manuscript found on Wikipedia. But it's nice because it reminds us how traditional this is. Okay, how old, what a venerable institution lecturing is. Okay, that if you went back actually to the 13th century Oxford and you walked into a classroom there, it would look very familiar to you. This thing that we're doing is very familiar. I was having a conversation with Bob Simmons this morning. I said, can we take this further back? And he said, yes, Aristotle, and used a word, peripatetic, that I'm going to have to dig into a little deeper. But yeah, so this is a very old tradition lecturing, okay? So what happened in the lecture part of the course? We had a textbook. Students were assigned a reading, okay? Then they come into class, and then I basically explain what was covered in the reading to them again. Okay, that's deliberate, you know, we're going over it again. You've got the book's understanding, now you get my explanation, okay? And then eventually there's quizzes and there's tests, and you know, it's very comfortable, very familiar. And then there was a lab course, okay? This is the cover of the lab manual that we used. It's about that thick, and somehow it cost $120. My students were kind of upset about that. 14th edition, so we've been doing this for a long time. What do you do in the lab course? We didn't do all the labs, we did a few of these. Well, you have a protocol. You have instructions, okay? You follow the instructions, you learn some things, how to use a microscope maybe, how to move small volumes of liquid from one tube to another, good stuff, okay? And then you get an outcome, or you see something, okay? You get a result. There's an expected result, okay? It's not new, okay? And it more or less complements the lecture. For instance, in the lecture, they might have read about mitosis, right? I might have lectured to them about mitosis. And then we might go and look at cells in the root tip of an onion, stain them, put them under the microscope, and they get to see mitosis, which, by the way, is really quite nice when you actually see it. It's, you know, if you let yourself be open to it, it's breathtaking. It's magical to see these chromosomes as things that you've stained in front of you. Okay? So this is how we do it, and this is what's often referred to in the literature. This is the traditional teaching method. Okay? And by the way, this is the way I had it. This is the way I had a genetics course. And I loved it. I loved this. I was great. Certainly never questioned this, and I think I learned a tremendous amount in both of these. But although, you know, this afternoon though, when I was looking at this, something did dawn on me. I can't remember a single thing from the lab I had in genetics. I remember some moments of lecture, I remember Janice teaching it, I remember the textbook we used. I can't remember a day, not an hour, not a minute of lab, which might be telling me something. Okay, so if we're going back to that question, you know, how should we teach biology? The way I taught it is probably one of the first thing most of us would hit upon, or rather the way it was taught to me, excuse me. Okay, it worked for me. I learned a lot, right? Ought to work for everybody else. Okay, this is the way I'm teaching genetics in 2018. It's a little different. The first version of this slide had arrows connecting the various things to the other things. They went away. They didn't last very long. This was totally incomprehensible with the arrows, so we're just going to have to make some links here, okay? I've color-coded things. In blue for you are learning goals. Okay, this is where you start in the design of the course now. This is what you want the students to learn. Okay, specific intended learning objectives are on the syllabus, right? So they know what they're supposed to take away. Learning proficiency, I'm going to talk more about that. That's the idea of learning how to learn this stuff, okay? Getting more capable at that. Technical competence, okay? I can use a microscope, I can do immunogenesis, I am comfortable working in the laboratory, I am comfortable working on the computer getting papers, okay? In brown here, we have the things that are designed so that the students are learning new concepts, new facts. In red are assessment tools. And in purple here are motivators. Okay? What's missing here? There's no lecture. There's no traditional lecture left in this course. That is to say, I try very hard not ever to get up and talk just to explain something most of the time. I have to qualify that a little bit. I do get up and talk quite a bit. Today I was up talking most of the class. But I think of this as inverted lecturing. Rather than me explaining, I'm up there framing the question. I'm posing questions. I'm guiding students to construct their answers to it. Okay? And I am explaining things which are a little solved, things which can help you, shortcuts that can get you to build your own conceptual answer sooner rather than later. Okay? But the lecture that I was doing in 2015, that's gone. Okay, that's essentially gone. The lab's gone. The lab's gone. Instead, we have there it is, research project, which we are working on. Okay? The dichotomy between lab and lecture is gone. Okay? We will be tested and quizzed on things that we're doing in lab days. When we are in the lab hours, we will be going over the literature. We will be doing all kinds of things. So the idea that these are separate modules, that you could have them be a standalone, that's gone now too. The idea is that this is now fully integrated. Okay? And we have all these motivators. So motivation reflection. Throughout a lot of the first week of the class, we talk about why we would talk about these things. Why genetics? Why are you here? Why is this a valuable thing to learn? Okay? Why might you be personally interested in learning this? Okay? The idea being, we're going to do better work if we know why we're doing this work at all. Okay? If we've actually asked and come up with some sort of an answer for that question. Okay? Pedagogy explication. I'm going to be talking more about this. This is the idea that if this is a little different, if this looks a little different than a course you've sat in before, you're probably going to do better if it's been explained to you why. Why is this a little different? Why are we doing the things that we're doing? Okay? So we'll actually, I don't know, I remember if I did it this year, but in previous years we've actually looked at papers that talk about this pedagogy, from the literature, talking about that. Collaboration. Students do a lot of work in groups, in teams. In fact, most of the work that they do in this class is done as group work. Okay? I'm not going to go through every little thing on here, but this is the idea about how the course has changed. Now, why and how? Why and how did we go from genetics in 2015, rather different, rather traditional, to the way I think of genetics in 2018? Okay? Does this work? Does this work just as well? Or better? What's going on here? Okay. So first off, well, why would you change? Why start this ball rolling in the first place? Is there anything at all wrong with that lecture lab paradigm? I loved it. I did great, I think. Well, we could argue that point. But I enjoyed myself, and I think I learned a lot. There's a couple different ways to come at this. I like this quote, and I'm going to read this to you. I don't recall when or how I stumbled upon this article, but probably secondhand through something else. But when I read this, it really, really resonated with me. I've been teaching maybe 4 or 5 years now. The approach to teaching anything that first occurs to most people is one I used for many years. Namely, I would think very carefully about the topic I wanted to teach. And after having figured it out very clearly, I would then explain it to the students so that they could understand with the same clarity. To my frustration and confusion, whenever I looked in any depth, it was clear that the students never understood the subject as I had hoped. There's a break, more things get said, then we cut to. However, after working in my research lab for just a few years, they were transformed into expert physicists. This is a physics professor writing this. Okay? Now this is just absolutely was my experience when I started teaching biology in 2005, because this is precisely what I did. Before I went into lecture, I would like run through this concept that I was teaching, xylem transport. I would just, like break it down. Like, what is the essence here? What's the fundamental thing? Water transport, osmosis, something like that. Just what is really fundamentally going on. I would think about it more deeply than I ever had before. And then I'd walk into the classroom with this argument, this structure of how I was going to explain that, which I thought was beautiful, ready, suitable for framing. And I'd give it to my students, and a few of them would give it back to me beautifully on the next exam, but many of them wouldn't. They had not gotten it at all. Okay? And I was frustrated and confused. I really was. I was like, what? I don't understand. I laid that out. It took so much time. We went through that. How did you not get it? Frustration and confusion. All right, so it's this, well, when we actually go into the teaching world, we sometimes, we get evidence that suggests this isn't working as well as we thought. There's another argument that's put out there. This is a different approach to this. So this comes from a paper by David Lopato. He's a colleague of ours at Grinnell, and he is doing a lot of work on science, STEM pedagogy, okay? He is sort of quoting an earlier report by, what was her first name? Tobias, in 2009, and it was called "They're Not Stupid, They're Just Different," I think was the name of the report, something like that. Okay. Can't remember her first name, but it'll come to me. One solution to the possible shortfall Tobias suggested was to recruit students from the second tier, a loose category of students who are able to do science, but for reasons of learning style, expectations, and experience, choose not to. Students of the second tier, Tobias wrote, may have different learning styles, different expectations, different degrees of discipline, different kinds of minds from students who traditionally like and do well at science. So the idea that's coming out here is that because I did well in lecture does not mean that lecture is great, it means that I'm odd, potentially. It means that I'm a little strange, okay? This idea of this second tier, what Tobias was really taking issue here was with this whole concept of introductory courses as weed outs. Okay, he really didn't want that. She said, well, we shouldn't be trying to weed people out. The idea is that there's some people who come hardwired, to do science. We're trying to identify those people and get the other ones out as quickly as we can so that we can devote our energies to these people. And she was arguing that's perhaps short-sighted, okay? And we don't want to be doing that. I was a little suspicious of this, particularly the learning styles that are not a thing that we subscribe to any longer. There was never any really good evidence for that. But, this second tier idea is interesting. I want to address this shortfall. So this was 2009 when this was first coming up. Okay, the shortfall is still with us. Most recently in 2012, the President's Council of Advisors on Science and Technology delivered a report to the President where they said over the next decade, we're projecting that we're going to get this many STEM graduates out of higher education with undergraduate degrees in science, mathematics, technology, engineering, and we're going to be about 1 million short of what we think we need to keep our economy humming. We need 1 million more. Go do something. Okay, that was the gist of it. So we're right in that window now. Okay, 1 million is a lot of students. All right. So there's this idea that the lecture lab model is perhaps not working for these people. That we can get more people on board. Okay. Another thing I'd point out, not on the slide here, is that in the lecture lab model, we're not teaching something. We're not teaching science. We're teaching biology, we're teaching a lot of concepts, we're teaching a ton about what we know. And we actually do teach, you know, I always taught a lot about how we came to know it, a historical approach to teaching it. But we're not really having them do science. We're not teaching them how you identify questions that are out there and then answer them. You know, find the unknown, the real excitement. So for those three reasons, we might sit back and say, well, we might want to think about some other things. Now, oh yeah, I'll go back to this. They were transformed into expert physicists. So this begs the question, what do you need to be an expert in your discipline? And since we're talking about biology, what constitutes biological expertise? So that would be the first question we want to address. This is how I break it down. Know stuff. Facts and concepts. Concepts being, you know, these overarching ideas about how we put knowledge together. Evolution by natural selection. Okay? Energy flows through ecosystems. Matter cycles through ecosystems. These are the sorts of big concepts that we have to get in there. When we talk about experts and how experts come at a problem, this is how experts do it. They come at it conceptually. Okay? They're reverting back to one of these great ideas, and then they're putting the information that they have into that context. Beginners don't do that. Beginners say, oh, what's the equation? Well, we don't have a lot of equations in biology, so what they normally fall back on is jargon, actually. They start throwing darts with vocabulary. Is it this? Is it this? You know? So we need to get those concepts in there, but you also need, frankly, a crushing number of facts. I mean, there's just a lot of stuff you gotta know. Okay? Know how to learn more stuff. You know, this stuff is changing all the time. This stuff's going out of date. I'm doing everything I can to make the textbooks in ethylene go out of date, and then we're going to have to relearn them. Okay? Learn more stuff. And this is mostly going to be off the printed page. That's how we do it. It's not mostly by someone lecturing to you. It's mostly by reading in the primary literature. Possess technical competence. Okay? When you pick up a pipette mint as an extension of your hand, do you not have to think to know how to use that thing and get a precise volume from one tube to another? Thank you, Audra, for all that you have done to make that a reality. Can you use a microscope? Can you sit down and just, you know, do you know how to use that thing, okay? So can you work with these stuff? Do you know how to find a paper in the literature? Can you go get it? Technical competence. Can you write effectively? Technical competence, okay? Communicate. Well, okay, communicate. I forgot I put that in. Craft questions. To be an expert, to be a real biologist, you have to be able to identify the cutting edge of what we know and what we don't know, and say, there's lots of questions out here. This one's really going to push us forward. This is the question. This is what we want to know. Okay. Now that one's in blue. That one's in blue. I'm going to come back to this craft questions thing, but I believe my 13 years experience has suggested to me that this is not where we want to put the emphasis at the undergraduate level. In fact, we might be making a real big mistake if we put a lot of emphasis there. This is all within range. This is all within their grasp. Crafting questions, that I think is what you go to graduate school for. Okay? I'm going to come back to why I'm making that argument. Just to keep myself honest, I went and made sure that this list was largely congruent with our biology department's student learning objectives. Well, we have the last one's not on there. We actually have 6. But here they are, and lucky me. I wrote this list honestly, I promise. I mean, this was like off my syllabi. But here we are. Become proficient in standard laboratory equipment, retrieve, synthesize, and critically evaluate the scientific literature. Know how to learn more stuff, right? So we're all on the same page here. Looks good. So this is the answer to the first question. And you know, all the lists, there are other lists, the National Academy has come up with lists of things. They all look basically like this. So this is our goal. All right. Let's go back to this question of how do we get from genetics 2015 to genetics, what year is it? Yes, genetics 2015 to genetics 2018. So I want to take you back to actually 2006 sometime. I'm a professor at William & Mary. And I taught my first plant physiology lab. Can you see that at all? It's good? Yeah. Sure. Yeah. And I decided I was going to do something different. I thought this was tremendously creative and innovative at the time. I wasn't going to teach a traditional plant physiology lab. Instead, I was going to take my 6 students basically into my lab, okay? And we were going to roll through specific objectives on my most recent NSF grant. And they were going to get their hands dirty doing them, actually doing some work on them, okay? Now, why was I doing this? Not principally because I was thinking about active learning or anything like that. I was doing this because I was thinking, I have a problem. I have a heavy teaching load and high research expectations. I got a win-win here. My students will get to engage in real research, and I will actually get stuff done. Okay? I will actually perhaps have some data generated out of this. So, okay, this seemed very nice to me. So this integration of your teaching and your research. How did it work? Well, they did get stuff done. So these are the 4 students from Bio 419, and they put together the gene construct. This is a translational fusion on a gene I was working on at the time called HAM1, and they made a HAM1 GFP, linked it to a jellyfish protein that fluoresces under light reporter gene, and I later gunned it into the plant, and there it was, suggesting that the protein is nuclear localized, and that work was done by Kristen Bishop, who was not in the class, but who picked that up as an independent research project. Kristen got her PhD at the University of Illinois about 2 years ago, and since I was here, I got to go out and see her. It was fun. She's now working for the federal government. I said, "What are you doing?" "I can't tell you that." Oh. This worked. At least, stuff happened. They actually got stuff done. I wasn't thoroughly assessing this in terms of, how is this affecting student learning. My main concern was, are we actually, getting stuff forward? Am I teaching and moving my research forward at the same time? But it was great, okay? About this same time, or in this same interval, I got an email from the National Science Foundation from a program officer there named Terry Wooten. She's since retired. And she invited me to come be a panelist to review grant proposals in a program that was then called TWOS. The program has since been cut and it revives, it comes back under new names. TWOS standard for Transforming Undergraduate Education in STEM or in Science or something like that. And when I was there doing that, Terry gave me this. She gave me my first copy, my only copy still, of Vision and Change. So, are there best practices for teaching undergraduate biology? Apparently, yes, there are, and they're all in here. Okay? This was published in 2009. There's a very, very long list of authors in the back of this thing. So this really represents a consensus view, right? And there are 25, count them, action items in this thing. Things that we need to be, I know, 25. I was somewhere recently, what was that, after we just had our Armistice Day, and I was reading that when the French Prime Minister was told that President Wilson had 14 points, he said, 14? My God, God himself only has 10. They've got 25. Here's just a few of them, okay? Action items. Engage students as active participants, not passive recipients in all undergraduate biology courses, okay? And if you go a little deeper into this, they're really talking about that active learning, that student-centered learning paradigm. That's what we're going for here. Ensure that undergraduate biology courses are active. Here it is again. It just keeps coming up. Outcome-oriented, inquiry-driven, and relevant. Okay? Now you have to interpret all this a little bit, but I admit. Facilitate student learning within a cooperative context. This is another theme that comes up again and again and again here. We're seeing better results when students are working a lot with other students, okay, when you're actually doing things to facilitate that, okay? And this one I put in red because this is mostly what I'm going to tell you about today. All of these things are in that Genetics 2018 that you all saw, okay? In fact, most of the 25 are in there somewhere. This one in particular I want to talk about. Introduce research experiences as an integral component of biology education for all students, regardless of their major. Not doing the regardless of their major part at this point, but getting it in there for sure. Okay. So I found my best practices. Okay. We start putting these into practice. So Not too long after that, I got a job at Rollins College. This is experiment number 2 from 2013, 2014. And I had my idea for the lab. It was bigger and better than it was before. Instead of taking students into my research area, which at the moment was, to be perfectly frank, a little non-existent, I was going to come up with a research project instead that was designed specifically for the lab, for the course. Okay? This was customized for them. Okay? And it was going to be a continuing project. The idea was, 1 year students would start on this, they'd make some progress, they'd leave it off at a certain point. We'd write a poster, it'd be on the wall, the next class comes in, they pick it up where they left off. Okay, so this kind of goes on. Okay. This is my first class doing this. Fuda should not be touching her skin with her glove. I incessantly harp on that. And they're busy working. So how did this work? I was very fortunate. I had a colleague in chemistry. His name was James Zimmerman, and he was paid by Rollins. He got course release and all this for this to be the pedagogy guru, at least for the sciences. Maybe he was for the whole campus. Okay, so he was your go-to guy. And one of the things he would do is he would come into your class and he would do these assessments where he would sit down, you leave the room, and he actually talks to the students, asks them some questions. They get to talk among themselves. They talk to him. He kind of filters it through. Okay, so I had James do this. How did this work? Well, they were working on a project to do with desiccation tolerance, and I'll just cut to the chase. It was not a great success. All right. So this is from James's write-up on this. Was this course a valuable and/or enjoyable experience? Explain. Valuable. We learned how to improvise and how to set up our own experiments. Not enjoyable. We felt the laboratory was quite unstructured and unorganized. The time we spent talking about hypothetical processes, period. Okay, that's in sentence fragments. Most of the time we found lab incredibly frustrating. Okay, I went home that night and I don't remember, I had a couple scotches and I watched the movie Gettysburg and just tried to I was pretty upset. So this didn't work as well as I'd hoped. What went wrong? What went wrong? So, try to evaluate this. Well, 2 things I decided. 2 things probably. Okay. Thing 1, what are they talking about here? Hypothetical processes, what's going on? Well, what we spent a lot of the lab doing was me trying to get them to come up with questions. Trying to get them to formulate the research question. And they were just struggling with that. They were completely struggling with that. And the more I thought about that, the more I thought, well, yeah, of course they were struggling with that. I've been thinking about plant evolution and plant physiology and all these things, for the better part of I'd rather not tell them. And they haven't. They're taking their first course in plant physiology, I'm expecting them to fly, and they don't have enough of a base to really formulate that research question. And that's what they were telling me, I think. There was another thing wrong here too, I think. I walked into this and I started teaching this lab, and then I walked out and the lab was done. I never explained to them why we were doing what we were doing. I never told them, this is the idea here. This is the concept. I just said, here's what we're doing. But without any explanation of why this was going on. I think that's why this kind of failed. So you pick yourself up, you dust yourself off, and you come back in 2014. So I threw that research question out, completely out. And we found another one. This is the other one. And what was magical about this was that it was all around us. So we're in Florida. We're just north of Orlando in a little suburb called Winter Park. And this is the resurrection fern growing on an oak tree. Okay, and this stuff's everywhere. You just see this stuff. It's on all the oak trees. And this resurrection fern right now looks thoroughly dead. I mean, it's crinkly, it's crackly, it's 15% of its wet weight. I went out and took this picture because there was a forecast that we were going to get a lot of rain that night. And I came back in the morning, put my camera in the same spot and took a picture again. That's what the resurrection fern does. This is absolutely fascinating biology. Not least because we have no clue how they do this, really none. There's some papers on it. They've got great titles like, such and such protein may be associated, yeah, right, well, yeah, great, thanks, with this phenomenon. So we don't know what's going on here. So the research project, that's Bitsy, you can see how pleased she looks to be working on this. And this is a gametified picture that they took. The research project was first just Can we take this organism into the lab? Can we culture it? Can we get some DNA out of it? Can we do the things we would have to do to work with this? It worked very well. So James came back the next year and asked, there's a lot on here, and I just highlighted in purple the parts that matter to us. What aspects of this course and or the instruction helped you in learning most deeply? How have these aspects helped you learn? The labs are really interesting. We're making progress. Also, we understand why and what we are doing in the lab, not following a recipe every single lab session. So they seem to be kind of jazzed about this. We're allowed to go into depth in lab because it is a little bit open-ended. And then James is in parentheses there. Yeah, okay, that's James in parentheses. So they liked it. They liked it a lot, actually. Okay, I got some of the warmest feedback to this. So those two changes were made. At the outset, I told these students what was going on, what was going to happen, what this would look like, that we would not know the answer, that we would come up against walls, that we would be troubleshooting things, that they would be confused, and they just needed to embrace that. And they were great with that. They were fine with that. Great group of students. And I told them, what we were doing and what the question was. I didn't let them try and figure that out. I said, here is desiccation physiology. And we looked at some papers about it. We read through some papers about it. We all got on the same page. And I said, our question is, can we work with this organism in the lab? Okay, let's go to work. Okay, so they were given the question. All right, and it worked. It worked very well. All right, so unbeknownst to me, I had not invented anything. Actually, in the first year I got here, I got an email from Frank saying, hey, there's this conference happening at this university in Texas, Austin. You might want to go down there, and I did, and I discovered that this exact thing I was doing had a name, and many other people were doing it in various places, and this was what was called CURE, course-based research experience. Okay, I've already defined it for you. Why is this a big deal? Why are people doing it? They place authentic research experiences within reach of all undergraduate biologists. This isn't such a big deal maybe for us, okay? Because we're doing this already, we're small. But at large schools, larger universities and things, this is absolutely key. The idea is everyone can get access to research. If you're at a larger school, it's very hard for everyone to get into the lab and learn by the apprenticeship model. Authentic research experiences are encountered earlier in students' careers when they can be more impactful. Okay? And societal benefits. Increased college retention, we think. Increased completion of STEM majors, we hope. Learner benefits, enhanced learning for the students. Teacher benefits. The same research teaching synergy that I was talking about before can be going on there. Challenges. You need a lot of stuff. You can't do research on any old things you happen to have lying around. You do need state-of-the-art equipment to be able to do state-of-the-art research. Okay. So that's what we're doing. Now, does this work? Well, there's literature on this. Literature takes two forms. One is quite a few papers by David Lapata mostly that are surveying students. It's asking students, well, what do you think? Did you like your cure? Did you learn more? Do you feel more like you're part of the scientific community? Do you feel more passion for the discipline, okay? And those suggest, yes, students like this, okay? They are student reported though, okay? Can we get an independent measure of how effective these are? That's really a hard thing to do, because we just generally don't have the conditions for that. We don't have this student taking this cure course and the other student having the identical experience, except they're taking a different course, right? In the same university. Well, actually, we got something like that in 2016, okay, from a study, and Erin Dolan was the lead on this, and she was the speaker, the main featured speaker at this conference I went to. So she was instrumental in getting this program going at the University of Texas at Austin. It's called FRI, Freshman Research Initiative. And a significant number of the students at the University of Texas, they all have a science requirement, okay? A good chunk of them, we're talking big place, right? Thousands of students. A good chunk of them did it through a traditional lab, and a chunk of them did it through this FRI, okay? The FRI is a cure lab. The way it works is In your first semester, you take what we call research methods. In your second semester, you get on a research project, okay? And you do that under close supervision of a postdoc usually. They're at the university, they got postdocs. Your third semester, you work much more independently or with some other students. And they did this comparison because really they had this, now they've got this under control. They've got students doing it one way, they've got another set of students doing the other way, okay? Well, is it helping you get through college? Graduation in 6 years? Yes, it was, okay? This is a statistical bump in the FRI cohort relative to the non-FRI cohort in this paper. Is it getting you to stay in STEM? Okay, yes it was. Okay, they were getting more students staying in STEM majors, not leaving them to go to other majors who were doing the FRI. Was it improving your GPA? No. That wasn't happening. Okay? So what do we make of this? It's interesting. And one real thing I want to point out is this is about it, by the way, folks. This is what we got. This is the data. This paper is what we have that looks like this, where we have something like a control and experimental. So far, this and David Lopato's student-reported data is what we have to go on. Okay? But it's interesting. There's a lot that's not here. We don't know what component of this rather complicated experience was leading to these gains. Okay, we really don't. And that's a question that we'd like to try and suss out. But this is encouraging anyway. So let me answer some questions for you, and then if you'll indulge me, I'll take just a couple minutes and I'll tell you about what we're doing now. So what do we want biology undergraduates to learn? These things, which we already talked about: effective communication, learning how to learn, learning the concepts, proficiency in the lab, okay, notebook keeping, all the things of research. Crafting questions for authentic research may often be too ambitious. Okay, this is no longer my goal. Okay, I'm no longer putting any emphasis on this. Okay? I'm prepping them, and they'll go on to grad school, and that's where they'll learn how to do that. That's where I learned how to do that. I realized I was not doing this when I was an undergrad. I didn't craft the question for my senior thesis. Are there best practices for teaching undergraduate biology? Yes. They're in Vision and Change. And they involve inquiry, active learning, and collaboration. Okay? Student buy-in to unfamiliar learning paradigms is critical. If you are changing things up on them, you have to get them on board. Okay? You really do, or this doesn't work. And I think that's what I learned doing this at Rollins. Okay? You've got to get that buy-in. How do we create conditions for undergraduate biology students to fall in love? Well, the answer I'm pursuing is authentic research experiences. Okay? The concepts are beautiful. I find them beautiful. I find them lovely. They're elegant. They explain so much. But research may be a more effective way. Research puts you in a larger community. We're social animals. We like working with other people. We like thinking, I'm now a part of a group. I'm now a part of a community, okay? Research is exciting. It's personal. When you discover something, you have some ownership there. I got that. That's my page. I'm naming that thing, Yeah. And I think this is more of a hook than learning about the concepts that are everyone's and outside of us. CURE provides a mechanism for incorporating authentic research experiences earlier and more broadly relative to summer research or independent research, these other ways that we can go about getting this. And they're great. I'm not knocking them. They're wonderful. Okay? Okay. So, what do? So, I came back in 2016 and I said, okay, we're doing a CURE lab. It's great. We're going. And I'm working on ethylene. So we're working on ethylene, people. So we asked this question: can we employ forward genetic screening to elucidate the cellular basis of ethylene perception in this fern, Ceratopterus ricardii? Okay? I won't go into the details of why this is such a profoundly fascinating question, okay? But we got right to work on it, and in 2016, they showed that the ferns do respond. However, they're responding at a very late stage in development, and when they're very large, and this is not good, because we need to do a genetic screen, which means looking at a lot of them while they're still little. So we came back the next year and said, look at the roots. And they also respond. In orange here we have the root length in 10 part per million ethylene. In blue we have air. It's a significant response, and it's nothing that you'd ever see with your naked eye. It's nothing you could quickly screen. It's not suitable. So we answered this question: no, we can't. And that was 2 years. and we're done. Now this is supposed to be an ongoing project, so we're going to have to switch gears here, and that's what we did. And I'm going to skip through a little bit of background here, but we're still working on plant evolution, okay, very important. And I still have this age-old problem, which is this is how we view plant evolution typically. This is from Science Made Stupid. The evolution of plants is an important chapter in the history of life. However, it's a pretty dull chapter, so we'll skip it. So we got to try and get rid of that first. So we're actually very interested in this, okay? You'll pardon me, I'm going to skip through this. This is a paper that inspired us, okay? They're looking at these structures. These are called rhizoids, okay? They're single cells. And they're anywhere that you see plants with gametophytes, okay? This is a liverwort, a very distantly related plant to this plant. This their roots are Arabidopsis thaliana. And it doesn't have rhizoids, it has root hairs. But what this paper showed was that a lot of the same genetic machinery that you need to make a rhizoid is in fact also playing a role in making a root hair. And it suggests that these are not different things. These may in fact be the same thing, just popping up in different places in evolutionary history and on different tissues. Which is actually really quite interesting, okay? Because the evolution of rhizoids I think is extremely important in plants being able to get onto land, okay? We realized with our ferns we could take a whole different look at this because ferns are rather special. They have rhizoids and they have root hairs. Most plants don't play that game. Very few do. Ferns do. So we could mutagenize ferns. We could look for gametophytes, small, easy to screen, we knew we could find things here, that are defective in their rhizoid growth. And then we could ask a very interesting question. We could say, if you are defective in your rhizoid growth, are you also defective in your root hair growth? This is much more compelling evidence that root hair and rhizoids are in fact the same thing. We're not looking across genomes separated by 420 million years of evolution. We're looking in the same genome on the same day. So we thought that was elegant. I certainly did. And that's what we got to work on. And we're working on it now. These are two of the students who are in the class now, Sam and Madelon. They're busy screening, okay? We've had lots of bumps, and I would tell you more about them, but I have expended a lot of time. But we've found some interesting things we were not looking for, okay? Typically, the rhizoids are about 1/5 of the length of a thallus. My students came to me 2 weeks ago and said, I think we have a phenotype because the rhizoids were substantially longer than we normally see. We do not think that this is genetics, however. We think it's this contaminating fungus that got on our plates. That's our best guess of what's going on. But this is nonetheless not only interesting, but potentially useful. Because if we could find conditions where we could grow rhizoids that are that long, and then look for gametophytes that don't have rhizoids, they're gonna pop. They're gonna be out there like a searchlight. We're gonna have no trouble finding these things. So we're pursuing this. It's this little gift that landed in our laps. Or we would be pursuing it if we weren't in the third week of November or the second or whatever we got. But Madelon's going to pick this up. She said, hey, I'm on this. I want to do independent research. I want to keep this going. Okay? So we're getting students excited about this. Okay? Moving forward, we're going to need some more things. Dissecting scopes, microscopes. I've already tried once with the NSF with a lot of help from Chris. We're going to try again. The TOOS program died. This is what replaced it. And we're going to try and get some bucks to support this. And then finally and lastly, the entire biology department, we had a little retreat, I don't remember when, some time ago. We had donuts from Casey's and coffee and we sat around. And we decided to apply to join Seepages. Seepages has been going on for about 10 years now. We're cohort 11. We were invited to join. Okay? It's an extremely exciting program where you get on board with this research on phage diversity that is spearheaded out of the University of Pittsburgh, Graham Hatfeld's lab. Okay? This is a paper that's published on this. This is David Lopato doing work again. And this is again, we're surveying the students. What do you think in regards to research process, knowledge construction, readiness for research? How do you feel about that? Well, in green here is cure. forgot to put the key up. In blue here are summer research experiences. And in red, that's seephages. Okay. They are consistently saying, I'm getting more out of that than I am out of these other pretty darn effective ways of learning about how to do science. So we're doing seephages now. That's Sarah in the foreground there. She's tilting the tube so that she can get the lysate that she's just put down into the cells, and the phage can go pop, okay? Phage, by the way, I have not described or defined for you. They are viruses of bacteria, is the way we typically think about them, okay? And we know so little about them, and they are the most abundant biological entities on Earth. By a lot, by a whopping number. And yet they're essentially a black box. Well, our students are helping to characterize that black box, to fill it in. And so far it's going real well, okay? All right, so I have already taxed your patience and exceeded my time a little bit. Thank you. Some acknowledgments. All my research students of all the different stripes. So I'd like to say thanks to, particularly you folks for coming today. Thank you very much. Thank you too, Nick, even though you're an ILA student. Some of my colleagues in particular, Chris Fasano has been terribly supportive of this. He helped us with a neutron experiment, neutron mutagenesis experiment I didn't have time to tell you about, and he helped me a lot with that grant proposal, and I'm sort of counting on himself for the next one too. I'll just throw that out. David Timberman was really very, very supportive when we came to him and said we want to do this seephages thing. He didn't even say what's that going to cost. He said, okay, great, let's go. So thanks for that. And since he left, Mark Wilhart has been helping us a lot too. And all my colleagues in biology, I mean, this is a biology initiative. I'm not doing this. I'm doing this with James and Tim and Ken and Kevin. And Tim and I went out together for the training last summer, had a blast at the University of Baltimore doing this. James, Tim, and I are going out to HHMI headquarters in Chevy Chase in December to learn how to do the annotation, and we're going to bring that back. The second half of the lab is bioinformatics for our students. We're very excited. Monmouth, thanks. The HHMI, thanks. They're the people who run the Seephages protein program. And the NSF for getting me out there to do that panel, because that was really my introduction to this world. I don't know why they picked me and sent me that email, but it certainly was transformative. So thank you, and I'll take some questions if we have time. Thanks.
Choosing the questions, and so sometimes you're trying to come up with active research questions that will engage the students through the entire semester while introducing them to lots of facts and technical competence that haven't been done before. How do you get the questions?
That's a really good one. So let's go back to the last question I got. I'd already used that paper that launched that Han Cannon paper. We'd actually already talked about that in genetics class as an example of doing a screen. And it was a fairly approachable paper. And it was a paper that mirrored a lot of the concepts we were talking about in class. So building off of that, you know that they have a lot of conceptual framework for that, okay? The other beautiful thing about that question was that it has a very simple answer. You do a screen. And the ethylene stuff, which integrated with my research and had that bonus, right? I'm getting work done. There were too many moving parts. I really think so. There were just too many things going on. We had to grow the plants in a strange way. We had to figure out how to mutagenize them. You know, it's too much. One of the ways you learn to do research is by doing something over and over and over and over. You start to get really good at this. You also start to understand why you're doing what you're doing. So the new project, tell me in the back row there if I'm wrong, but I believe it's fairly repetitious, right? I mean, once we get going, we do it again, and we do it again, and we do it again. And that's what research is like. So you're getting that experience, but you're also getting that ingrained training. So that was a criteria in choosing that question. The Ferns in Florida, they were all around us, so the students were walking around campus and they'd see their question. I thought that was kind of nice. I mean, it really makes it real and interesting, you know, getting them to think about what's in front of you there. Whereas the previous year, it was much more abstract. These were plants they'd never seen before. So all those sorts of things go into it, but it's a bit of an art form, I think. I did go over a little on this.
One quick question.
Yes.
Has the integrated lab lecture space enabled you to, I mean, is the lab you're teaching in one of those lab lecture that this building is supposed to have? Is that been beneficial or?
Do you mean, do we have lab and lecture in the same physical space? We don't, I wish we did. That would be nifty, because then we could sort of, and I have occasionally gone into lecture and said, alert everyone, we're running down the hall to lab because we need to do a thing. That's happened a couple times. No, we don't do that though.
So you don't have a.
No, and that's one of the challenges. Okay, the registrar is not thinking this when they set up the schedules. They're thinking traditional lab. Okay, when this building was designed, the infrastructure that was put down, the height of the benches, the absence of gas, they were thinking traditional lab, and they're great for that. If you're switching gears, you have some challenges. In the phage lab, we got all the students working like this, because that's where the gas is along the wall, and we need that to do the work. It's a little suboptimal, but you find ways to make it work, and we're getting it done. Yeah.
Just as one who fought for gas, you're lucky you have any at all. So, believe me, there wasn't going to be any gas in those labs. So,
Thanks.
yeah, none. So,
Bob.
So you mentioned that one of the things that you thought would work great, but ended up not working so great, was at William & Mary having a research project of your own to which students were contributing in the class that they were taking. Are you integrating your own research projects into the teaching you're doing now? I mean, or is the teaching you're doing now leading to research that can turn into publication or presentation?
But now it's going the other way. So yeah, the first 2 years of the genetics lab, that was that was absolutely out of my research. We were working on ethylene. We were trying to fill in an evolutionary piece there. But I decided not to do that again because, kind of like as Michael was suggesting, I mean, if you really want this to shine, if you really want it to hum, you got to get the perfect question. And maybe your research doesn't generate the perfect question for an undergraduate ongoing lab. So let's get that. It's still an evolutionary developmental question. It's still a fascinating question, and I'm still prepared to get enough data to publish a paper on that. After a few years of this is the idea. So now that teaching component is its own research project. And then other students who want to do independent projects, maybe building off that. That's what Caitlin and Daniel and Maddie were all doing. They'd all taken the lab and they'd said, wow, okay, this is fantastic. Let's just keep doing this for independent research. And they took the skills that they learned and they went to the next question. That's what Madelon's going to do now, and maybe others will join her. With the phage lab, I mean, none of us were phage biologists. Right, James, you're not phage biologist. DNA guy. So, we are now though, because we're all doing this, and we're all contributing to this project. Our names also go on with the students' names on the publication that goes into GenBank when we publish these genomes. And moreover, the hope is that the students who do this, they've got this set of research skills now. They can keep doing phage research, and they're freshmen. So they got 3 potential years to put these skills to use if they want. And we can mentor them, and we've got the space for it. And so we're hoping, yeah, research comes out of that. So we're phage biologists now. Yeah. Which is a lot of fun midway through your career, Yeah.
How's the love and how do you measure it?
Scientifically. You don't. I mean, you don't measure that. I mean,
People who go on to grad schools,
people go on to grad schools, people write you letters, people send you notes, people send you emails that say things like, this made a difference for me. I didn't think I could do this, but it really worked. I've got a card in my office from a student who took genetics in the 2nd year. It's just such a sweet card. It says, "Your class certainly gave me migraines, but I must admit, it really made me learn. I learned more in there than I had in any other class I'd ever taken, migraines accepted." So it's that kind of stuff. I had an email from a student last semester who she actually came to me and said, "I'm worried about the final." I said, "What's the problem?" She said, "I don't know how to study for it." I said, "What's the problem?" She says, "I don't know what to study because we covered all this stuff. I feel like I know it." Well, perfect. I mean, that's exactly what we're trying to do. So it's that kind of feedback. But yeah, I agree. I mean, I'd like to put the love meter on too and see how we're doing. Thanks for your patience.